जेल इलेक्ट्रोफोरेसिस के बाद हमें जेल की प्रिपरेशन की ज़रूरत पेश आई थी बिफोर वी कैन प्रोसीड फर्दर फॉर ट्रांसफ़र. Similarly, the membrane has to be prepared before it can be used for transfer of the DNA onto the membrane. Normally, the nylon membrane or any other membrane is available as a large sheet in the form of a roll. So we have to cut the size of the membrane required as per the size of the gel we have run for DNA samples. It is recommended that the size of the nylon membrane should be 1 millimeter more from each side as compared to the gel, which can be done using a fresh scalpel blade, or it can be cut using the scalpel blade or the scissor, which is meant only to use for the nylon membrane. Then 2 thick filter papers of the same size as your membrane is there. The filter papers are also going to be 1 millimeter larger in size on all the 4 sides. While handling the membrane, extreme caution needs to be taken. Blunt end forceps is suggested or recommended to use to handle the membrane, because if you touch the membrane and if the membrane gets some oily impression on it, oily dots on it, then the membrane will not get wet and subsequently the transfer cannot be done on this membrane. So for transfer purpose, after cutting the membrane of the required size, we have to float it in a dish containing deionized water. The membrane is going to get wet. After that, shift the membrane to appropriate transfer buffer at least for 5 minutes. Using a clean scalpel, we have to give a cut mark either on the top right side or on the lower left side as we did with the gel, so that the 2 cut marks can match together and we can place the membrane over the gel. अब अगर इस तस्वीर को देखें, इस तस्वीर में एक समरी की गई है हमारे पास जेल की. जेल के ऊपर हमारे पास नायलॉन मेम्ब्रेन के ऊपर फिल्टर पेपर 1 एंड फिल्टर पेपर 2. अगर आप इनके टॉप राइट कॉर्नर्स को देखें, they all have a cut mark that gives us a reminder of the orientation of the DNA, and we will have an idea where is our well number 1, where is our well number 2 samples, and where is our well number 3 sample. So once we prepare the membrane, we can proceed further to establish the tower that looks like this for transfer of the DNA from gel to the membrane through capillary transfer method.